Assalamualaikum and good morning. My name is Nur Faranis binti Mazlan, student ID 2023670648 from the Applied Microbiology program. My supervisor is Dr. Muhammad Shafiq Azmi. The title of my final year project is Comparative Study on the Antimicrobial Activity of Leaf, Flower, Stem and Root Extracts of CLITORIA TERNATEA against BACILLUS SUBTILIS and ESCHERICHIA COLI K12. My presentation will follow this outline: introduction, literature review, methodology, expected outcomes and challenges, Gantt chart and references. First of all, let's talk about the background of my study. Antimicrobial resistance, which is known as AMR, is a growing global health threat by reducing the effectiveness of life-saving treatments and placing populations at heightened risk. To find new antimicrobial agents, we turn to medicinal plants as a vital source of structurally diverse bioactive compounds like CLITORIA TERNATEA, that commonly known as butterfly pea or locally bunga telang. It is a perennial herb used locally, for example, in the making of nasi kerabu. My study will use two model bacterial strains, which is Bacillus subtilis, a gram-positive bacterium, and Escherichia coli K12, a gram-negative bacterium. They represent different cell wall structures, which is a key to this comparative study. This brings us to the problem statement. C. Ternatea is widely recognized for its medicinal potential and promising sources of antimicrobial agents. However, despite increasing interest, there has been limited evaluation of its antimicrobial potential. Existing studies predominantly focus on the leaves and flowers only, leaving the antimicrobial potential of other plant parts, particularly roots and stems, underexplored. Consequently, it remains unclear which plant parts have the most potent antimicrobial potential and whether the related bioactive compounds differ significantly across the plant parts. In addition, most available reports provide limited comparative analysis against different gram-positive and gram-negative bacteria, which limits the current insight into their spectrum of antimicrobial activities. To solve this, I propose three research questions. The first question is, do extracts from the leaf, flower, stem, and root exhibit significant antimicrobial activity? The second one is, which plant part produces the most effective extract? The third one is, are there significant differences in activity against different gram bacteria? My objectives is to determine the antimicrobial activity of leaf, flower, stem, and root extracts of C. Ternatea against B. subtilis and E. coli using the well diffusion method. The second one is to compare the inhibitory effects of different plant parts of C. Ternatea in terms of their effectiveness against gram-positive and gram-negative bacteria. The significance of study is threefold, which is the first one is to add systematic and comparative data for a plant common in Malaysia. The second one is it identifies the most bioactive plant part to guide future research. The third one, it supports fundamental microbiological principles by providing data on bacterial differentiation. For the literature review, the key concepts are clear. The enormous and irresponsible use of antibiotics has led to antibiotics resistance. We must explore complementary and alternative sources for novel antimicrobial agents. C. Ternatea extracts contain major phytochemicals such as alkaloids, saponins, flavonoids, and anthocyanins, which contribute to their uniqueness. The previous study summary shows evidence. This table from a 2020 study shows the inhibition zone of methanolic extracts of C. Ternatea leaves against various microorganisms such as Streptococcus agalactiae, Salmonella typhi, Staphylococcus aureus, Enterobacter aerogenes, and our model Bacillus subtilis and E. coli. This confirms activity but only for leaves. This leads to the knowledge gap identification. While previous studies have proven bioactive compounds exist, most research solely focus on the leaves and flower part only. The ability for stem and root part remains unknown. Therefore, my project aims to investigate and compare the antimicrobial activity for each part of plant extracts. Now, let's move on to the methodology part. The first part is the preparation of methanolic extracts. My experimental workflow starts with plant collection of C. Ternatea authenticated by a botanist. Each part is thoroughly washed with distilled water to remove any dirt and debris, followed by drying various plant parts in a hot air oven at 40 to 45 degrees Celsius until constant weight is achieved. They are then grounded into fine powder and stored in airtight containers. For extraction, the powder for each part is soaked in methanol and stirred using a platform shaker. Finally, the extracts are filtered using Wetman filter paper number 1 to obtain methanolic extracts. The next steps of methodology involve microbial work. I will prepare standard media such as nutrient-agar, Mueller-Hinton-agar, and Mueller-Hinton-Broth. The bacterial cultures of B. subtilis and E. coli K12 will be provided by faculty. I will obtain a pure culture using the streaking method confirmed by tests like morphological observation, gram staining, and biochemical tests such as catalase test and oxidase test. The core experimental workflow used the methanolic extracts. Antimicrobial tests will be conducted by using agar-well diffusion method to address objective 1. For objective 2, minimum inhibitory concentration, which is MIC, can be determined using broth microdilution method. All data analysis will be done by using two-way ANOVA. For the key materials and media include nutrient broth, Mueller, Hinton, agar and broth, methanol, Whiteman No. 1, filter paper, B. subtilis and E. coli K12 culture, each plant part such as leaf, flower, stem and root, and 3% of hydrogen peroxide for catalase test. My variables and parameters are defined. The independent variables are plant part such as leaf, flower, stem and root, and the second one is bacterial strain. The dependent variables are zone of inhibition in millimeter and minimum inhibition concentration value. For the key parameters are, the first one is incubation period, such as 24 to 48 hours. The second one is density of media, which is 0.5 McFarlane. And the last one is incubation temperature, which is 37 degrees Celsius. For experimental design and replicates, all experiments will be conducted in triplicate to improve data reliability. Results will be expressed as mean with standard deviation, and statistical significance will be analyzed using two-way ANOVA. Moving on to expected outcomes and challenges. The expected outcome is to identify the most potent part from C. ternatea with the strongest antimicrobial activity and minimum inhibitory concentration, which is MIC, against both bacteria. This supports microbiological principles by providing comparative data on the differential bacterial strain in various parts of plant extracts. I am aware of study limitations. Plant-derived compounds may exhibit pro-diffusion, resulting in a smaller zone of inhibition. Variability in phytochemical composition may occur. There is a potential risk of microbial contamination. Thus, I prepared a mitigation plan, which is all plant materials will be collected from the same location and processed under standardized conditions. All antimicrobial assays will be conducted in triplicate in order to improve data reliability and reproducibility. And lastly, strict aseptic techniques will be practiced to prevent any contamination during experiment. My Gantt chart details the timeline across Semester 5 and Semester 6, covering 11 key research activities from identifying a proposed title to the submission of the final report. This table shows the plan duration for each task, ensuring the project remains on track. I provide you the key references that have been used for this slide. Thank you for your attention. I am now ready for your questions.